Well, thank you very much for the invitation. I'd like to thank Professor Natarajan and the program committee. Since I did not know whether which session I will be invited to, and I just noticed yesterday, so I just modified the slides so that it will fit mostly into these sessions. So I'm not an ophthalmologist. I am a molecular biologist, molecular genetist, I actually say, from the Tokyo Medical Center, which is a national hospital organization, which is a Japanese hospital under the Ministry of Health of Japan. So we do a lot of basic research. We collaborate with many clinicians and try to identify the disease-causing genes and mutations. So today I'd like to focus on the retina, and of course this session is on the macula, so I will be talking about the macula. So macula is a very unique tissue in the eye, and this structure is very unique. This structure only exists in higher primates and also very few type of birds. And so that's why the experimental animals like rats, mice, dogs, cats, pigs, cows do not have a macula. Only higher primate with a high resolution vision have a macula. So what kind of risk factors is involved for this macular diseases? And one factor is, of course, genetic factors and also environmental factors. Environmental factors contains like blue light, obesity, smoking. These kind of risk factors is also environmental factors, but also there will be genetic factors. It is a little bit difficult to measure the environmental factors, so we usually try to start with the genetic factors. So that's what many people have done for age-related macular degeneration, as we heard from the previous speaker. So this is called the Manhattan plot. You see chromosome 1 to chromosome 22, and it is showing that the association of a DNA, which part of the chromosome of DNA sequence is actually associated with AMD. This is a comparison of a control, 30,000 people control versus the 30,000 AMD patients, and it's called the Genome-Wide association study, and this is called the Manhattan plot. It looks like. So as the lines goes upper, it means that the p-value 10 to the minus over 10 to the minus 8 is being considered as an association. So among these genes, you can see on the chromosome 10, there is one place that is highly associated with AMD. The p-value is 4 times 10 to the minus 353. So this is one of the places that we have to study. So that's what we did for this. Here, when we zoom into this chromosome 10 area DNA sequence, there is 3 genes that are highly associated with the onset of AMD. So this line, red line, is exactly what the sequence is. So we focus on this part of this regions to see how this can lead to age-related macular degeneration. When you sequence this part and compare with the controls, you would see that this red part of these 2 genes is changed in the AMD patients. So what does that mean? So this is a experimental procedure that we do. We take the normal patient's DNA sequence, and then we change to AMD patient's DNA sequence and measure the promoter activity. Promoter activity is the amount of expression of RNAs from this DNA sequences. So number 1 is the control AMD, control person, and if you replace the AMD sequence, you can see that the transcription is increased significantly. So it means that, I'll go back 1 slide. It means that if you have a AMD sequence here, it means that the HTRA1 gene right behind this sequence, the transcription will go up. So this is what is happening. This is a normal DNA sequence, and then usually you will have a suppressor protein that is binding to this regions to suppress the HTRA1. But if you have an AMD sequence, one of the suppressor is lost and replaced by an activator protein. And this increases the transcription of HTRA1, means more RNA, more proteins. So this is a cell that's called the iPS cell. This is a stem cell that you can make from each individuals, and you can measure the amount of HTRA1, whether AMD people actually have a higher RNA levels. So this is the AMD patient, this is the normal patient, this is the glaucoma patient, and this is the AMD patient. As you can see, if you have an AMD, HTRA1 level is higher in these people. Where is HTRA1 expressed in the eye? So this is immunostaining, and you can, if you can focus on the green part, this is where the HTRA1 is expressed, and this is mainly in the photoreceptors. So what we did is that, okay, in the humans, HTRA1 is highly upregulated. So if you make a transgenic mice that overexpress HTRA1, would you get a phenotype like human AMD? So exactly what we did is like that. And we use a special camera for mice, and then we do the fluorescent angiogram and ICG to see that, yes, there is a capillary coming out through the Bruch membranes into the retina. And here we can see that, yes, there is a CNB coming out. Although the mice does not have a macula, but you can replicate without the macula that the CNB can be replicated by just overexpressing HTRA1. So now we're currently collaborating with the pharmaceutical companies to suppress HTRA1. If it's high from the stem cell level, if you suppress with drugs, you will probably suppress the onset of age-related macular degeneration. So now I'd like to move on to the more extensive genetic factor diseases. And these genetic factors can be like macular dystrophies, Stargardt diseases that are caused by a single mutation in the gene. So we have many these inherited retinal diseases, and there's like about 36 inherited retinal diseases, including macular dystrophies and Stargardt diseases. These we call the IRD, inherited retinal diseases, is caused by almost 260 genes. It has been, first gene has been cloned in 1988, and now we have about over 260 genes. And as we analyze more population around the world, I think we will be about 10 times more. We will have more disease go. So I'm currently the head of the Japanese consortium, started in 2011 with 30 ophthalmology departments in Japan. And we are currently trying to solve this 36 Leber congenital amaurosis, retinitis pigmentosa, Usher syndrome, Stargardt, macular dystrophies, and so on and so on, retinoschisis, Best disease, so on and so on. And try to find. So we are currently in the center, and there's a clinicians sending out phenotype information and saliva or blood sample to us. We do the sequencing and transcriptome, proteomes, and gene editing to create a mouse model and then primate models. We are also in association with many foundations and societies in Japan, and developed a registration system, genotype-phenotype database, that the blue part, the clinician will input their OCTs and all this image fundus photos, images you can upload into this system, and the genetics will put all the genetic information into this part. So we have tried to increase the quality of this phenotype by asking this specialist, a disease leader, as a disease leader, to monitor the incoming data from all these universities of different diseases, if this is really that diseases. So this quality check is very important for this to run the consortium. And once it's cleared, and it goes through the whole exome sequence, whole genome sequence to do the whole thing. So we have collected about 1,300 pedigrees, 2,000 samples, and then we have sequenced the entire gene, 20,000 genes of all the patients that we had. So this is the whole exome sequence. And if it's a recessive family, we will sequence parents, affected, and even a normal brothers or sisters to filter the cause. Sorry. Okay. Oh, time's up? Okay. Okay. Okay, so in 2001, when we started the consortium, the detection rate of these mutations were 17%. But with this new technology of next generation sequence, the computing, everything, we have now increased just in 7 years to 53%. The Moorfields detection rate is about 65% currently, which has about 30 years of experience. So I think with this new technologies, any nations can quickly catch up to the most. So we are currently identifying the genes and try to find the mechanism by using a protein work, the iPS cells, knock-in mice models, and try to find a drug. So I would just show you two macular diseases. One is called occult macular dystrophy. This is identified by Yozo Miyake, the director of our institute. If you do a fundus photo, it's normal. The fluorescence angiogram is normal. And if you do a full ERG, it's normal. But if you do a focal ERG, you can see that the fovea cone is totally lost. So we took 3 years to clone the genes, and it's called RP1L1. We now have 50 pedigrees of this and confirming that this is the real genes. And we're trying to make a monkey model of this. This is another gene called LRRTM, leucine-rich repeat transmembrane 4 proteins. This protein is actually expressed in bipolar cells. Bipolar cells, it's a synapsis protein, but if it has a mutation in this proteins, the photoreceptor degenerate. And as you can see, this autosomal dominant family, you can see that the huge hole in the macular regions. So it's a very special gene, and also it's a very new phenotype that we're seeing that the bipolar cell dysfunction actually degenerates the photoreceptor, and it's specific to the photoreceptors. And we have been studying other genes. This is a new gene for Leber congenital amaurosis, and also there's a new gene for RP and rod cone dystrophies. So all these new genes is coming up, and I think if we go into more of the Asian population, we will explore more new genes that has not been found in the Caucasian population. So I will end with this slide that we have also started a global genetic consortium based on the experience of the Japanese consortium. Now 30 universities, countries are on board, and we will have this GEGC consortium immediately after this session at Hall H. So if you're interested in genetics, please come to us, and this is the website. So thank you very much for your attention.
I think any questions? We have quite an exciting field now. I think we're looking at his presentation like future we are looking at something.
Thank you for the presentation,
Thank you.
sir. I was curious as to what's your thoughts on using CRISPR-Cas for genomic editing for the inherited retinal dystrophy?
Yes.
And which one of those do you think is a good target?
So for gene editing, it's still is ongoing. I think it's a research, and then the recombination rate is about 5%, and you can improve this by up to maybe 10%. But I think still you can recombinate safely about 10%. But also you might have a off-target which will replace and damage other genes, and it's you have to be very careful with this. I don't think there is a safe technology for it. We should wait for another technology to come.
You did mention that you're using therapies. So which ones, what therapies are we developing? If you could
Yes.
elaborate
So
on that?
basically, we're testing through this metabolism changes. Especially, we will find a lot of mutation that will enhance the interaction of protein with usually not wild type. Only the mutant will bind to a specific proteins. So in that case, if you have a drug that will prevent from binding, I think it's a very, and I do have another slides for those, but yes, so drug development is of course good. And also for the recessive families, putting the normal cDNA into it should help significantly. But for others, especially for dominant families, I think gene editing will be the future, I think so. Yeah.
Yeah.
Thank you.